Okay, this is your DNA notes. This is for chapter 13. You should have gotten the page from the chapter 13-14 drawer that says DNA notes. Some of this you've gone over before with our bookwork, and you took a quiz actually already on this. So some of this is review and some of this is new. So stick with me because the new stuff is in the second half. So remember, DNA is our carrier of our genetic information. It's going to hold all the information to tell a cell what it wants to be and what it needs to look like. We look the way we do because of our DNA. Remember, your DNA tells you everything about you. So DNA is really important. And this is one of those things that makes everything different. So we need to know what's going on with our DNA. We should know not just what it looks like, but what it does and how we use it, which is what we're learning in this chapter. So remember, what organelle holds DNA? Well, it's the nucleus in eukaryotes. And it's the prokaryotes that don't have a nucleus, so their DNA is going to be in their cytoplasm. And remember, which one are you? I say you weird because you are a eukaryote. So we have our DNA in our nucleus. A prokaryote, that's like bacteria, they're going to be out in the cytoplasm. DNA is made of nucleotides put together. A nucleotide is the monomer of DNA. If you don't remember that from chapter 2, you might want to write that to the side of your notes. It is the monomer. A nucleotide is the monomer of the nucleic acid group. A nucleotide has 3 small parts, a phosphate, a sugar, and a nitrogen base. You can see the picture over here, phosphate, sugar, and a base. I call it circle, house, rectangle. That helps you remember it. They're going to come together to form one nucleotide. Now, on your notes, it asks you to draw and label this picture, so it's right here. You might want to pause and draw it. The coloring's not as important as the words, so make sure to pause and draw that. There are 4 different options of nucleotides. I included their pronunciations in case you're not sure how to say them. So the first one is guanine. The second one is cytosine, the third one is adenine, and the fourth one is thymine. Most of us want to mess with that ending and make it 9, like guanine, and I get that because that's how English works. But remember, these aren't English words, these are actually Latin words, so that messes the ending. So now it's guanine instead of guanine. But we also can shorten it to just the first letters most of the time, to G, C, A, and T. And when I see G, C, A, and T, it reminds me of this. And this is probably the weirdest thing I've shown you all year. But when I see that, I think of G-Cat. I think of some gangster cats hanging out in their hoodies with their chains on, listening to their cat rap music. I don't know. But it helps me remember. And not just that, if you know from the notes and from our bookwork, G and C go together and A and T go together. So G-Cat, it's funny, but it helps you remember the 4 bases, and it helps you remember who pairs with who. G with C, A with T. Now we're going to talk real quick about something totally new, purines versus pyrimidines. Purine and pyrimidine. A purine is a base that has 2 rings. That's adenine and guanine. It does not mean they pair together because they don't. They're just the 2 that have 2 rings, and I'll show you in just a second what that looks like. A pyrimidine is a base that has 1 ring, and those are cytosine and thymine. However, a purine is going to pair with a pyrimidine. They never pair with each other. So remember, A goes with T and G goes with C. So the purine adenine goes with the pyrimidine thymine. Now when I say rings, this is what I mean. I'm talking chemistry. A purine has 2 chemical rings. A pyrimidine has 1 chemical ring. That's all I mean by that. But a purine goes with a pyrimidine. So A would go with T, G would go with C. These nucleotides always come in their pairs. A with T, G with C. And remember, do you remember the guy who talked about that? Who figured out that pairing? We're going to talk about him in a minute. Remember the DNA we colored? Here's a picture of it. It says on your notes that you need to draw this, so I really do want you to draw it. Again, the color is not as important, but the DNA itself is. It's going to show you that A and T go together. It doesn't matter which side they're on. It's always A with T. So T with A with T, whatever. C with G, G with C, and you can see it's because of the way they fit. Now this is not exactly what it looks like in real life, but it's to show you what we mean. See how A is rounded? It wouldn't fit in the pointy end of G. it's only going to fit in the rounded match of T. It's like a puzzle piece. They fit together. So this never changes. They're always going to pair this way because it's how they fit. So once again, go ahead and pause the video and draw this on your notes. Coloring's not important. You can color it if you want to, though. So here are some of these scientists. We've talked about them already back in our book work, but we're going to write about them real quick. So some of our scientists, Watson and Crick was one. They figured out the shape of DNA. So they're the ones that actually called it a double helix or a twisted ladder. Rosalind Franklin took an X-ray image of DNA, and this is the picture that helped Watson and Crick with the shape. Now I'm going to go on a tangent for like a minute and a half. Stick with me. It's a pretty interesting story. So Rosalind Franklin took the first picture of DNA, and Watson and Crick used this picture to make their model of DNA. Okay, this happened in the 50s. So Rosalind Franklin worked for Watson and Crick, not because she wasn't as smart as them, but because in the 50s, women had to work for men still. We weren't allowed to have our own labs. Well, Watson and Crick made this awesome model out of the picture Rosalind took, and they won the Nobel Prize. That's a huge prize that you win when you do a really cool discovery. They didn't mention Rosalind Franklin on their discovery. They pretended like she didn't help at all. They took all the credit themselves. That always makes me pretty mad, because I think she deserves some credit, and I get really frustrated that they didn't share it with her. And Rosalind Franklin didn't really say anything. And guess what? Back in the 50s, we didn't know a lot about X-ray protection. If you've had an X-ray before, you know that you get covered with a lead vest, especially over your reproductive organs, because X-rays cause mutations. Well, Rosalind took so many X-rays of DNA to try and get that picture for Watson and Crick that she got cancer. And she died because she took this picture, because she spent so much time with X-rays. So not only did she get snubbed for the Nobel Prize, but she died. And Watson and Crick did not. Later in life, the Nobel Prize Committee, posthumously, that means after death, gave Rosalind Franklin the Nobel Prize that she deserved with Watson and Crick. But it always makes me a little bit mad that they didn't share it ahead of time. Our last guy, oh, sorry, I forgot about these pictures. Here's a picture of Rosalind and then Watson and Crick. Fun fact, Watson, this little goofy guy right here, he's still alive, and he lives in Chicago. If your heart wants to, go ahead and Google him. He's got a very interesting Wikipedia page. A little bit of drama in his life. It's James Watson, if you're looking for him. Here's our last guy, though, Erwin Chargaff. Everybody wants to call him Chargaff because we're American, but he is not American. He was, I think, French. So his name was Erwin Chargaff, not Chargaff. Erwin Chargaff is who figured out that A and T and G and C base together. He did this because he separated the DNA all out. You can do that with chemicals, do a bunch of chemistry. And he saw that the amount of A, he got the amount of adenine bases, equaled the amount of thymine bases. And the amount of guanine bases that he pulled out equaled the amount of cytosine bases. It wasn't just a coincidence. He figured out that these matching were because they matched, because they paired together. So they should have the same amount. Now, when we talk about how they pair, they're pairing using chemical bonds. Remember, we talked about how you can bond things together like water. Well, bonds join our DNA together. So there's a bond in the middle of the ladder. So between the A and the T, or between the G and the C, they're matched. So there is a hydrogen bond. It's a weak bond, but there's still a bond holding it together. Now, on your notes, this is just a quick practice. I want you to add the other side of the DNA. So if this has an A, that should be T. T with A. G with C. G with C. Keep going the whole way. When you get that one, you have a second one. Same thing. A to T, A to T. Down here, that T would go to A. So match those DNAs. If you want me to check them, just call me over and I will. Now, sometimes, this is our new information, sometimes our cells need to make more DNA. We just talked about one of the best times that a cell might need to make more DNA. Do you remember? Well, it's mitosis, right? When we make new cells, then we need more DNA. So in order to make that DNA, we're going to have to do something. we're going to have to go through replication. And replication is when we make a copy of our DNA. Do you remember the specific phase when we replicate our DNA in mitosis? It was in interphase. It was the S phase. So up here, and when we need to make more DNA, you should write in the cell cycle, specifically in interphase, the S phase of interphase. So when we go through the synthesis part of interphase, that's when we make more DNA because we're making a second cell. If DNA tells us who we are, then every cell we have needs to know who we are. So every single cell needs this DNA. So we have to make a copy. Replication just means to make a copy. Now think about what that word copy means. A copy is the exact same thing as the original. When I made these notes, at least the paper ones, I made copies. Everybody's notes should look exactly the same because they are the same thing. It's just a copy. So when we make a copy, a replicating piece of our DNA, we are literally making the same DNA twice. And there are 3 steps we're going to go through. So the steps, the DNA gets opened up. And again, I'm going to show you this in just a second. The DNA gets opened up. Number 2, free nucleotides come in and fill in the empty spaces. So in your DNA, you have nucleotides, right? Those are letters. Well, in your nucleus, in your cell, you have free nucleotides, just random letters floating around. And we use those to build DNA. They actually come from the food we eat. So when we eat food, it all has DNA in it. We break down the DNA into the individual letters and then rebuild it to make us. So the DNA gets opened up. Step 2, the nucleotides come in and fill in the empty space. And then the cell checks for errors in the copying and puts the DNA back together. You need to add that to your notes. And puts the DNA back together. That's not in the notes, so you need to write that down. They put the DNA back together. We're going to practice replication because we're going to actually do this with DNA. So here are the steps. You're going to draw this. So take it one piece at a time. You're going to take the DNA. Now it doesn't have to be this fancy. You can just draw a line with the letters. You don't have to actually draw it in all these boxes. Here's your original DNA. We're going to open the DNA up. This is step 1. So see how it gets opened and pulled to the sides? Then step 2 is right here. The left side gets a new right side, and the right side gets a new left side. Do you see how I went from green to yellow? The green is just showing us where the old DNA is. It's showing us where the original pieces go. The yellow here are the free nucleotides that came in. So here's step 1, we opened it. Step 2, we're filling in the left and the right side. Step 3, we check for mistakes, and we close it back up. Do you see how this top piece is exactly the same as this bottom piece? Now we're not looking at the colors, we're looking at the letters. So we have A with T, C with G, G with C, A, T, T, A. The second one, same thing, A, T, C, G, G, C, A, T, T, A. So from the start to the finish, we have the exact same letters, now we just have 2 of them. You've copied your DNA. Your notes ask you to draw this, and it's drawing it with the color. So make sure you do that. DNA is copied so the cell can go through mitosis. Remember, each new cell needs DNA, and even though we just said it's a copy, it's not an exact copy because there is technically an old piece and a new piece. But it's pretty darn close to being exact. So it's exact in the information, but it is an old piece with a new piece. Now there's 2 spots on your notes to practice replication. Here is the first strand you're going to replicate. You're going to do step 1 where you open it, step 2 fill in the sides, and then step 3 close it up and have 2 new pieces. You need 2 colors. Maybe you'll use your pencil or a black ink for the regular, and then for your new, go get a colored pencil or a marker or just a different colored ink. This is the one you're copying, so you'll want to pause, do that practice. Now it says have Miss Haverlack check your example before you move on. You want to make sure you're getting this right. You want to pause until you're done and then move to the next one, which will be in just a second. Here's the second one. All that changes down here. Look at this, it's different. You're going to replicate that on your notes as well. Have me check it again, because this is the new stuff. We're practicing our replication. If you get confused, then back up to this part of the notes and take a look. If it still doesn't make sense, then I'm in the process of making a video that will show you this in a different way. So if you're still not sure, I'll have a link to that video on the DNA replication practice assignment. Okay, that's the end of these notes. You do not have to turn them in. You shouldn't turn them in because you're going to need them to do the next assignment. If you have any questions, you can ask me. You can read your notes, check the book, or watch this video again. You have all the tools you need to do this.